All right, in this video, we'll be looking at the origins of cell compartmentalization. So how did these different compartments of the cell membrane and how did these different organelles arise in eukaryotic cells? Again, just a reminder what compartmentalization is, this division of the eukaryotic cell into separate compartments so that different sorts of metabolic practices and things can occur. And for just a comparison between, I don't have a picture for this comparison, but that's going to be okay. Move me out of the way there. Prokaryotic cells, like bacteria, are just single-celled organisms, and there's no internal membrane-bound organelles, whereas eukaryotic cells, which is basically all other life forms, have this sort of structure internally. Both cell types have a plasma membrane that separates them from the external world, and bacteria even have a cell wall that separates them. But there's much more going on inside of a eukaryotic cell than inside of a prokaryotic cell. Prokaryotic cells have an internal region called the nucleoid region, where the DNA is located, but a eukaryotic cell has an actual nucleus. So that is a protected area from the rest of the cell, so that particular nucleus things can happen. So looking at how this occurred, first we'll talk about so the nucleus, and I'll just go back here actually for this one. Nucleus and the internal membranes, like the endoplasmic reticulum, are theorized to have formed through enfoldings of the plasma membrane. Again, all of it ends up kind of connecting to the outer membrane eventually. So the ER here, the Golgi here, notice they're separate from the plasma membrane, but you can imagine at one time this plasma membrane kind of folded inside. They're all made of phospholipids. It's all the same material. So you could see how that could eventually have happened. But there's a couple of other differences there. Some of the different organelles have some different origin stories. For the mitochondria, the idea is that they evolved from previously free-living prokaryotes that were engulfed by a previous cell. So you have an aerobic prokaryotic cell, that an aerobic means able to breathe oxygen, was engulfed by an anaerobic cell through endocytosis. And so the engulfed prokaryotic cell, and rather than getting ingested, they formed a kind of a mutually beneficial relationship, whereas the anaerobic cell gave that small little cell protection, and the aerobic cell gave that bigger cell that it was trapped by, gave that bigger cell energy, and so they had a mutually beneficial relationship. Over time, the engulfed cells lost some of their independent functionality and became the mitochondria that we know and love today. Chloroplasts had a similar sort of origin story. They evolved from previously free-living prokaryotic cells through endosymbiosis as well. This idea of endosymbiosis is endo means inside, sim together, living, bio, and so inside together living. It's kind of like you get the idea that there's this free living photosynthetic cell that was prokaryotic. It was engulfed by another cell, and that engulfed prokaryotic cell did not get ingested, but rather they formed another mutually beneficial relationship. And over time, it lost some of its functionality, similar to the mitochondria, but in this case, it gave this entire cell the ability to trap sunlight and create sugar from it to do something like carbon fixation, allowing to take CO2 from the outside world and make something useful out of it, like sugar. There are various evidences as to why we think this happened. Both mitochondria and chloroplasts have a double membrane structure, as we've talked about before. So I can just kind of move me here, peeking out of the bottom. You can just, that's kind of weird. I'll just put me over here. Okay. So mitochondria and chloroplasts have a double membrane. So you kind of get the idea that their own membrane and then the previous cell membrane that engulfed it, they're together. This functions to regulate passage of materials in and out of the cell, gives it a stable internal environment. And so double membrane also adds to surface area. Like prokaryotic cells, mitochondria and chloroplast both have DNA, and it has a circular kind of DNA as opposed to the linear DNA that you find in eukaryotic cells. And they both have their own ribosomes and make their own proteins, and they reproduce independent of the rest of the cellular parts. And so the mitochondria and chloroplast will reproduce even if the rest of the cellular parts are kind of in a time of rest. They will reproduce independently of the rest of the cell. And so this is several evidences that suggest that perhaps mitochondria and chloroplasts were at one time independent, free-living prokaryotes, and now are a part of all eukaryotic cells in all eukaryotic organisms.